Welcome to another episode from the Veterinary Research Channel. Today, we delve into a fascinating study published in the Journal of Veterinary Internal Medicine in 2023, which investigates the impact of dexamethasone, a commonly used medication in equine practice, on the antibody response of horses to vaccination. Dexamethasone is a type of glucocorticosteroid well known for its anti-inflammatory and immunosuppressive properties. It is frequently administered to horses to manage various health issues such as asthma, allergic reactions, inflammatory bowel disease, and even certain types of cancer like lymphoma. While the therapeutic benefits of dexamethasone are well established, there has been limited understanding of how this medication might affect a horse's ability to develop protective antibodies after vaccination. Some earlier research has touched upon this topic. For instance, one study suggested that inhalant fluticasone, another type of corticosteroid, does not seem to interfere with the immune response to vaccines in horses. Another study indicated that a single dose of dexamethasone could lead to a decrease in the production of certain immune cells, specifically CD4 plus T cells, and reduce the levels of inflammatory signaling molecules in healthy horses. Furthermore, it has been observed that repeated intramuscular administration of dexamethasone to horses following vaccination against bovine diseases resulted in lower levels of specific types of immunoglobulin G, although the overall levels of immunoglobulin G in the blood remained similar to those of vaccinated horses that did not receive dexamethasone. Interestingly, the effect of immunosuppressants administered around the time of vaccination is also a subject of ongoing investigation in human medicine. Several studies examining the impact of corticosteroids given orally or through inhalation on the response to different human vaccines, particularly inactivated influenza and COVID-19 vaccines, have not consistently shown a reduction in the immune response in treated individuals. Currently, the Centers for Disease Control and Prevention have not issued specific recommendations regarding the use of corticosteroids before or after vaccinations in humans, although they suggest reducing their use in such situations whenever feasible. Equine influenza and equine herpes virus, one, are significant viral diseases that are widespread among horses. Outbreaks of these diseases can have severe consequences, ranging from life-threatening neurological forms of equine herpes virus, one infection, to substantial economic losses due to disruptions in horse trade, movement, and the cancellation of events. Therefore, ensuring an adequate immune response to commercially available vaccines is crucial for protecting individual horses and maintaining the health of the broader horse population. This study aimed to investigate the effects of clinically relevant doses of dexamethasone on the antibody response to commonly used vaccines against equine influenza virus and equine herpes virus types 1 and 4. The findings of this research could provide valuable guidance for the use of dexamethasone in horses around the time of vaccination. The researchers hypothesized that administering dexamethasone intravenously at the time of vaccination would lead to lower levels of antibodies 30 days after vaccination compared to horses that only received the vaccine. They also proposed that dexamethasone administration would result in a reduced systemic inflammatory response, as indicated by lower levels of serum amyloid, an inflammatory marker, compared to horses that received only the vaccine. To test these hypotheses, the researchers conducted a randomized study involving 55 healthy adult research horses. The horses were divided into 4 groups. The first group served as the control and did not receive any vaccine or medication. The second group received only an intramuscular commercial vaccine against equine influenza virus and equine herpes virus types 1 and 4. The third group received the same vaccine along with a single intravenous dose of dexamethasone, approximately 0.05 milligrams per kilogram of body weight, administered at the same time as the vaccination. The fourth group received the same vaccine and 3 intravenous doses of dexamethasone at 24-hour intervals, starting at the time of vaccination, for a total of 3 doses. The researchers measured serum amyloid A levels in all horses on the first day of the study, before any treatments, and again on the third day. Antibody levels against equine influenza virus and equine herpes virus, one, were measured on the first day and again 30 days after the initial treatments. For the equine influenza virus antibody measurements, a hemagglutination inhibition assay was used with a specific strain of the virus from Kentucky in 2014 as the target. For equine herpes virus, 1, a multiplex enzyme-linked immunosorbent assay was used to measure total immunoglobulin G and a specific subtype, immunoglobulin G47. The individuals performing the hemagglutination inhibition assays were unaware of which group each horse belonged to and when the samples were collected to ensure objectivity. The results of the study revealed that vaccination did indeed lead to a significant increase in the average antibody levels against equine influenza virus in the horses that received only the vaccine and in those that received a single dose of dexamethasone. However, this significant increase in equine influenza virus antibodies was not observed in the group of horses that received multiple doses of dexamethasone. Interestingly, there was no statistically significant increase in antibody levels against either total immunoglobulin G or immunoglobulin G47 for equine herpes virus, one in any of the vaccinated groups at 30 days compared to their initial levels. Furthermore, among the vaccinated horses, the levels of these equine herpes virus, 1 antibodies at 30 days were not significantly different between the groups that received dexamethasone and the group that did not. Regarding the inflammatory response, the researchers found that only the horses that received the vaccine alone showed a significant increase in serum amyloid at levels 72 hours after vaccination. The serum amyloid A levels in the horses that received either a single dose or multiple doses of dexamethasone at the time of vaccination were not significantly different from those of the control horses at 72 hours. There was also no significant difference in serum amyloid A levels between the group that received a single dose of dexamethasone and the group that received multiple doses. All horses in the study completed the trial and did not exhibit any clinical adverse reactions to the vaccine or the administration of dexamethasone. Initial serum amyloid A levels were within the normal range and similar across all 4 groups. Baseline antibody levels against equine herpes virus, 1 and equine influenza virus were also not significantly different among the 4 groups. The discussion of these findings highlights that dexamethasone appears to reduce the post-vaccine inflammatory response in horses, as evidenced by the lack of significant increase in serum amyloid A levels in the treated groups. Vaccinations are known to trigger an acute phase inflammatory response in horses, leading to elevated serum amyloid A levels for several days. The relatively short half-life of dexamethasone in horses after a single intravenous injection, ranging from about 18 minutes to just over 5 hours, might suggest a limited duration of effect. However, the prolonged suppression of the horse's natural hydrocortisone production and alterations in the concentrations of cortisol and certain inflammatory and immune-regulating molecules following a single dose of dexamethasone indicate that its anti-inflammatory effects can persist for up to 72 to 96 hours. This likely explains why the horses treated with dexamethasone in this study did not show an increase in serum amyloid, a level 72 hours after vaccination. In contrast to the clear effect on inflammation, the influence of dexamethasone on the antibody response to vaccination proved to be more complex. The antibody responses to the 2 different viral antigens in the vaccine, equine herpes virus, 1 and equine influenza virus, were not affected by dexamethasone in the same way. Antibody levels against equine herpes virus, 1 did not significantly change after vaccination, regardless of whether the horses received dexamethasone or not. However, the post-vaccination antibody levels against equine influenza virus showed differences depending on dexamethasone administration and even varied with the frequency of dosing. Horses receiving multiple doses of dexamethasone did not develop a significant increase in antibodies against equine influenza virus after vaccination, while those treated with a single dose or no dexamethasone did. These observations suggest that the way dexamethasone might modulate the equine antibody response is intricate and could be influenced by factors such as the specific type of antibody and the pattern of dexamethasone administration. The study vaccine contained inactivated, non-replicating viruses, which typically stimulate a strong antibody-mediated immune response by activating B cells and triggering the production of specific antibodies. While live vaccines can elicit an immune response through recognition of damage patterns associated with the virus, inactivated vaccines often rely on adjuvants to enhance their effectiveness. Adjuvants work by boosting the local inflammatory response and the release of signaling molecules called cytokines, which attract more antigen-presenting cells. This leads to increased uptake and presentation of the vaccine antigens and subsequent activation of B cells. Activated B cells can either quickly produce initial antibodies, mainly of the immunoglobulin M type, or they can develop in specialized areas called germinal centers. Within these centers, they undergo a process of maturation and switch to producing different types of antibodies with higher affinity for the antigen, as well as forming memory cells that provide long-term protection. Therefore, a local inflammatory response is generally considered crucial for a successful antibody-mediated immune response. This implies that dexamethasone could potentially reduce the immune response in horses by dampening the local inflammation triggered by the vaccine and by interfering with the signaling pathways and the processing of the vaccine antigens. Despite these considerations, the study did not observe a reduced antibody response against equine herpes virus in the horses treated with dexamethasone. Although the researchers did not perform detailed analyses of immune cell types and their functions, these results suggest that dexamethasone did not impede the immune response to the equine herpes virus antigens in this particular study. These findings align with previous studies in horses that also did not find a decrease in overall antibody levels after dexamethasone treatment. Furthermore, current recommendations in human medicine from the Centers for Disease Control and Prevention suggest that corticosteroid treatment can be continued when administering inactivated vaccines to people. The fact that no changes were detected in antibody levels against equine herpes virus in this study could indicate that dexamethasone had no measurable effect on this specific part of the immune response, or it could be related to the dose of dexamethasone used. It has been suggested that the effects of dexamethasone on the immune system might depend on the dose administered, although there is limited information on this in horses. Studies in humans have shown that a single intravenous dose of dexamethasone can lead to a dose-dependent decrease in certain types of immune cells, with higher doses resulting in lower cell counts. The dose of 0.05 milligrams per kilogram used in this equine study is common for treating various inflammatory conditions in horses, but higher doses might have different effects on the equine immune response. However, it's also important to note that antibody levels against equine herpes virus were not only unchanged in the treated horses, but also in the control horses that received only the vaccination. This could be because repeat vaccination of horses that have been previously vaccinated might not always result in a significant increase in antibody levels. Additionally, protection against equine herpes virus infection relies not only on circulating antibodies, but also on the cellular and local immune responses, which are not measured in this study due to the difficulties in quantifying them. Therefore, antibody levels in the blood might not be the sole indicator of protective immunity and vaccination success against equine herpes virus in horses. Interestingly, the horses in this study showed a considerably stronger antibody response to the equine influenza virus antigen compared to the equine herpes virus antigen. One study in thoroughbred racehorses even suggested that concurrent vaccination against equine herpes virus might enhance the antibody response to equine influenza virus without reducing the response to equine herpes virus. A similar interaction could have occurred in this study, although the underlying immunological mechanisms and clinical significance of this observation remain unclear. While antibody levels against equine herpes virus remain stable across all groups after vaccination, multiple doses of dexamethasone appeared to lessen the antibody response to the equine influenza virus antigen in this study. This contrasts with previous research in humans and horses that generally did not find a sustained reduction in antibody responses to influenza vaccines following different dexamethasone dosing regimens. The fact that horses still developed increased antibody levels against equine influenza virus even after a single dose of dexamethasone might suggest that any immunosuppressive effect of dexamethasone at this level was minimal and not easily detected by standard antibody tests. However, some reports in human and veterinary medicine have suggested that a single dose of dexamethasone could, under certain conditions, stimulate antibody production. For example, increased antibody levels were reported in a human patient with an enlarged spleen who received oral dexamethasone. In mice with latent herpes virus infection, systemic dexamethasone or acetylcholine treatment led to increased antibody levels, possibly due to reactivation of the virus and subsequent immune stimulation. Similarly, a study in catfish experimentally infected with a herpes virus showed a secondary immune response and increased antibody levels after a single intramuscular dose of dexamethasone was administered 90 days after the initial infection. If a stimulatory effect of a single dose of dexamethasone on equine influenza virus antibody production occurred in this study, it was not evident in the equine herpes virus antibody levels. It's unlikely that the horses were exposed to equine influenza virus during the study period, as none left the premises and no new horses were introduced. The differing antibody responses to the two components of the vaccine could also indicate that dexamethasone might selectively affect different types of antigen-specific antibodies. For instance, one study in rats showed higher levels of a specific type of antibody, immunoglobulin A, but not immunoglobulin G, after dexamethasone administration following vaccination. Whether similar selective effects occur in horses vaccinated with a multivalent vaccine requires further investigation. The researchers concluded that while the observations of this study might be specific to the horses and the vaccine used, they suggest that the effects of dexamethasone on the equine immune system are complex and not fully understood. Different dosages and administration schedules of dexamethasone or other corticosteroids could have varying impacts on antibody responses in horses, and these have not been adequately studied. Furthermore, this study primarily focused on the antibody-mediated immune response and did not investigate how dexamethasone might affect the quality of the cell-mediated immune response and the activation of T cells in response to the vaccine. Additionally, all the horses in the study had been previously vaccinated, meaning they had an existing immune memory. It is known that secondary immune responses after repeat exposure to an antigen are often stronger and involve memory cells. Immunologically naive horses might respond differently to corticosteroids given at the time of vaccination. Finally, the study did not include a challenge with the live viruses after vaccination, which would be needed to determine if dexamethasone affects the actual protection provided by the vaccine against equine influenza or equine herpes virus, infections, even in the absence of a significant decrease in antibody levels. Thank you for listening to the Veterinary Research Channel. A link for the referenced paper is in the video description. Remember to test your knowledge by answering the questions in the description, and as always, keep those minds inspired, hearts light, and tails wagging.